Okay, we'll try this again. Welcome, 7th period. We're talking about page 82 in your textbook, metabolic pathways. We talked about enzymes. Enzymes are controlling, right, these reactions. Metabolic pathways basically a sequence where this enzyme creates this product. This next enzyme takes that product and changes it to make it something else, and it continues on down the pathway. Are you with me? And they've given us a 4 enzyme pathway here, where you start off with one substrate, the enzyme changes it to make a second substrate, and it continues to move. Are you with me? Basically, it's like a recipe where it takes multiple steps to make your finished cake at the end. In this, there's something we call a rate-limiting enzyme, which is basically the start, the first enzyme. So in other words, if that enzyme decides, I'm done. I'm not going to make any more of my of my product. I'm not going to take any more of this substrate and make a product. It keeps then enzyme B and C and D from actually working as well. So it's rate limiting. It's the one that stops the reaction from continuing to work. It limits it. Are you with me? It's like in a group project, that one person who's really not paying attention and slows everybody down. Right? No, don't name names. I'm saying that's your rate limiting, right? That's the thing that slows it down. And again, all of this is up on Schoology, by the way. All these PowerPoints, everything is there for you. Okay? Shh. Any questions there? That's really the basic thing I wanted to see. You can get that these enzymes can stack up in this metabolic pathway. And the first one is the rate-limiting enzyme. Cool? I'm sorry?
How does it limit the rate?
It would just, because this guy, enzyme B here, cannot work until enzyme A creates its product. Because enzyme B is taking enzyme A's product and turning it into something else. It affects the rate because it can't keep going if this first guy is not doing his job, or if it slows down. Because B and C and D can't work until A does its thing. It's like a factory line, right? If you're building a car, the person who's building the frame, the foundation of the car, right, then passes it to the person who puts the seats in, who then passes it to the person who puts the windows in. Are you with me? But it all goes back to that first person. If they don't have that frame done, it limits what happens afterwards. Right. It limits how fast the end product is created. That's exactly correct. Okay? Shh. Stay with me now. All right, moving on. The next page. Talking about DNA and how it works. You remember DNA, of course. You've talked about this many times in the past. It is the genetic code, right, friends? Right. Thank you. The genetic code telling us basically how to make proteins. That's really the point of the DNA. It's basically the recipe book for all the proteins you need to function. All the different things that make the different cells in your bodies, they're guided by this cookbook, by this recipe book called DNA. And the gene is a portion of the DNA that contains information for one protein. So when we talk about a gene, that means here are the instructions of how to make, here's the recipe for one protein. Okay? Remember, DNA has 3 parts. The phosphate and the sugar are like the backbone, and then you have this base that's sticking on the front. And it truthfully is all about that base. About that base. No trouble. It's all about it, right? Just for you. So here...
I'm in a video!
It's classic. All right, so the nucleotides, that's what we call these guys, these bases. They come in pairs, right? They come in pairs. First, you have T and A that go together. T is thymine, A is adenine. You remember this from biology, right? C is cytosine, G is guanine. I never remember how to pronounce that one. Guanine, sure. This is how I remember it. TA and GC. Okay? That's how I remember it. Any TA or GC or even AT, Appalachian Trail. Okay, that's how I used to go for. But none of you all knew Appalachian Trail, so I went with Tacoma Academy. Okay? And these pairs go together, right? So making this DNA, you see here, the T and the A go together, the G and the C go together, and basically whatever is on this one side, the opposite end pairs it up, right? G's with C's, A's with T's,
Can we borrow the little
on down.
DNA 3D?
Little 3D molecule thing. I'm with you. I'm with you. I'm with you. We could try. We'll see. All right, moving on here. Because you know this. You know this piece. This is not me teaching you this the first time. All right, so let's talk about what happens when DNA wants to replicate. This is here on page 85 in your textbook. What happens is the DNA wants to copy itself perfectly, right? Any mess-ups, and we call that do you know what a mess-up in DNA is called? A what? Mutation, absolutely. This is what a mutation is. We've messed up, and somehow the translation, the copy has gotten messed up. Shh, stay with me. Stay with me. So therefore, we go through this thing called DNA replication. The molecule splits. You see that there on page 85 here. That there's actually these proteins that unwind the DNA. So that now I have two separate strands. And then we start to bring these base pairs to attach. They already have their phosphate and their sugar that's already attached to them. They're basically like the Legos for this DNA, right? And then they're going to start attaching on the bases, right? The T with the A with the T, the G with the C. And you basically end up with two DNA strands. Remember, this happens during interphase, right?
They're just like floating around.
Sure, they're in the nucleus there, and then that's interphase, right? And then very quickly then it'll move into that prophase and anaphase and metaphase, telophase, right? Where it sends that outside the nucleus. Nuclear wall breaks down. Remember all this we talked about? Okay? And then it splits, and one strand of DNA goes one way, the other strand goes the other way, and you have two brand new cells, right? Just going back here to make sure there's nothing. So again, the new DNA is one old strand and one new strand together, right? And you can see that in the picture here. This guy out here is our old one, and we're putting together the new one on the inside. Different kinds of mutations. There may be some that we'll talk about here. I truthfully can't remember. Good. Any questions there about DNA replication? Pretty straightforward. Well, let's see. What does your study guide tell you? What does the study guide say? Let's pull it up here so you can see it. The question is, how detailed do we need to know this thing? Let's go to chapter, it's not 3, it's 4. Chapter 4, study guide. And what does it say? DNA, you should know what the genetic code means. You should know what genes mean. You should know what genomes. Okay, these are all just definitions of what's in your textbook there. Shh. You should know the pattern of GC and AT, how they go together. So let's say I were to give you a string of DNA. Something like G, that's a C. G, T, A, C, G. Could you pair these up? I'm not sure quite what happened there. Can It's all right. Shh. Can you pair these guys up? What's going to go with the C? A G. What's going to go with this? Are you with me? So you could do that pretty simply, right? Shh. It's okay. Don't worry about it. Do you know how loud this is going to be on the YouTube video? I apologize, 7th period. I can't get these guys to shut up today. Stay with me, all right? I appreciate your comments, but stay with me.
Okay, well, I have a question.
Okay, yes, ma'am.
Why is there 2 G's in the bottom part of this 2 C's? Like there's CG.
Here?
No, a thing you just wrote. CGTAC, and then you get GCATC.
I'm saying that these guys are going to pair up,
Oh, got it.
and there will be a G here.
Got it.
Okay? And you should be able just to give me some information about how that works. I'm not looking for the super details. Okay? How it unwinds, bringing the base pairs in, you end up with 2 new strands of DNA. Good. Back to our PowerPoint. Genetic code, that again is this method of storing information for making proteins. Because really, the DNA is mostly about making proteins, right? So RNA molecules, these are the ones that actually do the replicating, the making of these proteins. So let's talk about that. If you remember, RNA have different base pairs, right? Instead of, well, you tell me. Instead of the T for the RNA, you get a U. You with me? A is still paired with T, but the U is paired with an A. That's the significant difference. Still G's, still C's, but instead of it being TA, now it's AU.
What does U stand for?
Uracil?
That's the base.
Mm-hmm.
Where's the base?
Yep. Uracil right here. What's that?
The pictures are inconsistent. Because the picture showed up before was like the base was the side.
I got you. No, phosphate, sugar, bases are on the end, the square one. Okay? RNA molecules are only one strand, not 2 strands. So no double helix, no spinning. It's just simply one strand of these bases put together. And what they do is they come in. They come into the nucleus, right? These messenger RNA molecules. They're in the nucleus, and they're the ones that are making this copy of the DNA. So basically, they're coming in, and they unwind this little portion of the DNA. Like this right here. Oh, boy. Don't go there. Let's try it again. They unwind this thing right here. You see it? Kind of like just parting the curtains, right? And they're going to copy just this one little portion here. That's what the messenger RNA is doing, right? Pairing up, if you have a C, I put a G. If I've got a T, I'm going to put an A. If I've got an A, I put a U for messenger RNA, right? Changes it. So that's my messenger RNA. That's inside the nucleus, right? And we call that process, I'm not sure if it's here, it must be on the next slide here. We call that transcription. Oh, it was on the last one. Transcription, right here. Wow. Sorry, 7th period, having trouble here. That's transcription. What does it mean to transcribe something? Transfer it, okay? To write it. A transcriptionist is someone who is like writing on a court case, right? Writing down all the things that are being said. Okay? So to transcribe means to write, which is basically what they're doing. We're copying it. All right. So we've made this copy in the nucleus. Now we're moving into what we call translation. Translation is now where that messenger RNA goes outside the cell. And where does it go to? Where does it go to make proteins? Nope. Remember back to chapter 3. What part of the cell makes proteins? The ribosome. It is right in the book there, right on the board as well, right? Okay? The ribosome is where it takes it to. And it starts this process that we call translation. So back to our picture. Number 2 here. And again, what page is this on in your textbook? Because it is there. Page 88, 87, depending on which book you have. 88 is where it is in the new book. Shh. The messenger RNA goes out of the nucleus and attaches onto one of these ribosomes. And the ribosome starts reading, basically seeing this little pattern of bases. And you can see that these 3 bases are called a codon. I'll point to them right here. Each pair of 3 of these bases is what we call a codon. And those 3 correspond to a certain amino acid. The example they're giving you here is that AUG is, I can't hardly read that, methionine. It's right there on the bottom of page 88. Okay? And each of these 3 little combinations means a different amino acid. It's like a code, right? If I give you these 3, you give me this amino acid. If I give you those 3, you give me this amino acid. And basically, this is going to combine all of these guys together to make a protein. Let's talk about a little more details. Yes?
Do you need to know
No, you do not have to know the specific codons and anticodons, no. But you should know what they are. That the codon is the piece of the messenger RNA that's out there. And the anticodon is what's actually on the part of the amino acid that's going to come and attach to it. Okay? Right. There's these things called transfer RNA that are bringing. Those are the little T-shaped guys. Right here. Those guys are bringing these amino acids to the ribosome. Okay? They're like the little, like the chaperone, moving that over to it. Okay? And then each of those little transfer RNAs has these anticodons. These, again, this little 3-letter, this 3-base sequence that attaches to that messenger RNA. Okay? Are you with me so far? I hope hopefully this is pretty straightforward for you. The ribosomes contain enzymes that join those amino acids together to make this chain, this polypeptide chain, right? And as I continue to join them, at the end you get this lovely, they call it a polypeptide chain, which we know as a protein, right? Here's a little zoomed in picture. Anticodon is this guy right here. That's that UGC piece right there. The codon is this number 5, the ACG. They correspond, right? The transfer RNA are these little T-shaped guys here. They're bringing the amino acid. Once they're in place, this peptide bond gets formed to bind those 2 amino acids together. Once the 2 codons are there next to it, the 2 transfer RNAs bring the two proteins together.
So transferring the anticodons to the messenger RNA helps create the bond.
Yes, sure. Yep. The anticodon matches up to the codon on the messenger RNA. And those proteins, those amino acids are attached to the transfer RNA. Once they get there next to each other, the bond is formed between them, and the transfer RNA moves away because it's done, right? It leaves. It goes away, and we're happy. And I'm going to keep doing this as long as there are messenger RNA codons Because that's the recipe. It's telling me what order of amino acids I need to build this protein. The tRNA would just leave, and it would go get another amino acid and come back to the ribosome to make another protein. Okay? I kind of think of it as like a chef. Well, a question, and then I'll go to my analogy.
And after the amino acid gets like left by the transfer RNA, do they like rejoin like directly after with the rest that were on the polypeptide chain?
Yeah, they're going to keep making this whole long chain here
No,
as
like after, like in the second to last picture when it leaves, does it
Here?
rejoin?
oh, this guy up here. You're saying?
No, not the transfer RNA, but the actual codons. Like, do they rejoin in a line?
I'm sorry. I'm not sure what you're talking about, Dominic. I'm sorry. Please, yes, come show me what you mean. I'm not getting you.
Like for this, after it leaves and it like goes like that, does this like rejoin like this, like how that is, and like that's how it creates the next side that's gonna intertwine?
I'm with you. So this guy over on this side here, this guy is bringing the amino acid
Okay.
to the chain. This guy on this side has already brought his. They've already bonded. He brought number 5 in here, right? And so now he's going to leave and go get another amino acid and bring that amino acid back to the ribosome to build another protein. This guy is coming to bond here so that this way 6 and 7 can bond together. And then another one's going to come here to make number 8. And a new one for number 9. Does that make sense?
Yeah.
They're coming in from the right, bringing these amino acids in this picture. Sure, yeah. From the view that we're at here. Yeah. After translation of the mRNA, I would assume it's going to restart again to make another protein. If it's needed. The same protein. It can only build one protein. It's just the recipe for just one protein.
But since it's already like has all the anticodons on it, like what?
Oh no, the anticodons would come off. And they're going to go get another amino acid. Yes, the anticodon stays attached to the tRNA. But it'll then go and get another amino acid to bring back to the ribosome. Exactly. All of this is done to make this lovely bond right there. Okay? Any questions here, friends?
So is it going from set, like the new ones are higher?
In this example, yes. The newer amino acids are the higher numbers, and the old amino acids are the low numbers. I'm just going to erase this, try and clear it off a little bit for you. Are you with me, my friends? Okay, I'm going to sign off 7th period. Once you please now, I'll tell you 5th period, the same thing I'm going to tell these guys here now. I want you to use this time now on your questions, and I'll answer questions while I'm here, but on the questions in your textbook, because those will be due on Friday. Okay, 7th period, I do want you folk to do that now. Working on those questions, studying this piece, because again, I want you to know how these guys work. Protein synthesis, DNA replication, and cellular respiration. Those are your 3 big pieces for the test. Study guide, no, I'm not going to have you fill all that out. I would recommend doing that. But the questions is what I will collect. Okay? Goodbye, 7th period.